So today we're going to talk about soil health. Now, Catherine has given me an excellent set of questions that she has compiled from many of you in terms of questions that you have provided to her. And so we're going to cover not all of those. If we covered all of those, we would be here for a very long time. So we're going to cover a section of those. Really what we're going to talk about is soil health. What is it? How we can measure it? What are some available tests of soil health? Soil testing, because you're going to be hearing about soil testing, and you've probably done some of it already in terms of soil health, or you have colleagues and other farmers that you know of that have done soil testing. And then we have a whole series of questions that I'm just doing rapid fire answers that we can elaborate on. Okay, and so I'm going to launch into it, straight into it, and here we go. The second slide, soil quality versus health. So I always like to start at this because let's say 25 years ago, the big thing was soil quality. The word soil health really wasn't used, and it was because there's a difference between the two terms. Soil quality is really discussing or tries to define the soil's ability to produce crops, okay? And whereas soil health does not necessarily relate, I shouldn't use the word relate. Its primary objective in terms of defining soil health is not yield, okay? So it's more the functioning of soil. How is that soil working ecologically, and how does it work compared to what that soil would have been, if you want to use the word naturally, or before agriculture, or before the management, okay? So two quite different terminologies. Soil quality is related with the ability of the soil, the capacity of the soil to produce crops, whereas soil health is the ability of that soil to function in the landscape and provide services, okay, in relationship to what it would have, its potential, if you want to call that, okay? So quite different things. It's interesting, over time, soil health has come to dominate in terms of our terminology, and we are thinking about, in terms of soil health, its relationship to crop productivity, of course. So they've been kind of smeared or blended over in terms of the definition. Move to the next slide. Why care about healthy soils? There's a whole bunch of functions of soil and a healthy soil. Decomposition of organic matter. We need the decomposition of organic matter to release nutrients from the plant material. Cycling of minerals and nutrients in soil so that the plants can obtain the nutrients that had been uptaken previously by plants. Related with that, it's a reservoir for nutrients to next crops. It's a redistribution of minerals and nutrients, and in other words, we want the nutrients to not just be in one zone in the soil, let's say at the surface, where we want the nutrients to be throughout the root zone. Sequestration of carbon, very important in terms of buildup of soil organic matter. This is probably one of the most important services that a healthy soil can provide, especially from a crop production standpoint.
Can you just remind me?
Soil structure in terms of improvement of aggregation, reduction of compaction, lowering of bulk density, water holding capacity, increasing it. And then degradation of pollutants and pesticides is a very important service of soil because in water, in air, even if you're not using pesticides or there's contaminants in the air from manufacturing and industrial processes and so forth like that, so those soil organisms will work to get rid of those contaminants. Biological regulation of pest species, and there are many other ecological goods and services such as improving biodiversity, water quality, and so forth. Next slide, traditional depiction of soil health. this is really basically the basis of soil testing for health. It's looking at the physical, chemical, and biological attributes of a soil. And there's a Venn diagram here where they have the overlap of the three circles. That really is about the target that we want in terms of assessing the soil health, okay? And primarily it's about structure and aggregation of a soil, okay? And not having compaction, ensuring there's good porosity for aeration, water movement, and water storage. And for chemical, ensuring that we don't have extreme acidification in our soils, that we do not have salinization or buildup of salts, and that there's good nutrient holding capacity and availability of nitrogen and phosphorus, particularly phosphorus in organic systems. And then there's the biological attributes of soil health, which would be macrofauna, such as worms, earthworms is an example, and there's microfauna, which would be nematodes, mice, springtails. Then there's microorganisms such as protozoa, bacteria, and fungi. And roots, we often forget that roots are vital biological characteristic of soils. Actually, in terms of organism amount and weight, roots are probably they are the dominant in terms of the mass of roots underneath in soil. We forget about that. And then there's just general biological activity of all those organisms working in concert that we refer to as the soil food web. And then there's organic matter, which is kind of a phenomenal measure of soil biological conditions. It kind of encompasses a whole bunch of different factors, so it's kind of like an umbrella soil property in terms of soil health. So all soil health tests will have a component of organic matter determinations. Next slide on soil health is central. How do we unmute?
I started my notebook.
So soil health is central, and this similar thing in terms of it's expanding a bit more. This is more holistic now, okay? Where we're talking here about farm health and talking about the sustainability of a system. It's a system, the farming system, is be able to cope with climate change, for example, or economic fluctuations and markets and so forth. Quality of the food, of diseases and marketability of the food, and then ecosystem health in terms of, kind of consider this also like ecological goods and services. So the benefit of that field in relationship to the landscape, and this we're really talking about here about air, water quality, and biodiversity in terms of with flora and fauna biodiversity that outside the farm fence and gate and so forth, okay? So bit more holistic, okay? Now this is you're not going to have the soil tests address these, okay? They're really not going to be addressing all of these. They're really just focus on the ability to produce crops, okay? What was that? 9-9.
The conference has been unmuted.
So I've just unmuted everybody and just checking in to see if anybody had a burning question or comment.
Yeah, it's true, I haven't. I just learned that soil health test. Does the Haney test apply to any of the soil health testing?
Yes, it does. The Haney test does apply, and we're going to briefly discuss about the Haney test, okay? And it's a very interesting test, and we're definitely going to go over that today. Excellent. Okay, I'm going to mute everybody again and then check back in about 5 minutes, okay? Or we're going to continue.
The conference has been muted.
So the next slide then, purpose of soil health testing. The little diagram at the bottom there really discusses shows the interaction. Soil management. So those are the practices that you would employ in the field, and that they relate to soil processes. In other words, how soil functions and works, and its benefits are good and bad in terms of promoting crop growth and also biological properties of soil. And then there's the soil testing, which tries to assess or measure, okay, the impact of that soil management, of those practices that we're doing in our fields, and that impact on soil processes, okay. So this is really important. Getting a report back is useless unless we kind of take some implications from it, okay? And a critical thing is asking, what am I doing to my soil? And then what is that, what I'm doing, how's it affecting how the soil works? And ideally we need help for that, and the soil health test, that's what they would be ideally useful for, is telling us the impact of our practices on those processes. The other thing about this little trilogy or trinity of testing and health is monitoring. Okay, we just don't want to do a snapshot in time. In other words, you just don't want to sample once and then say, okay, this is my health. That's going to give you some information, but not a whole lot. You're much better off if you sample repeatedly, test repeatedly. That will give you a better idea of where your field is heading in terms of soil health. Okay, is your management affecting or changing the way the soil works? Okay, so that's the important thing. So this monitoring is very important. And then if you just monitor with soil testing and so forth without adjusting your management, we're missing the boat. Okay, we're not capitalizing on the ability to evolve our production system. Okay, we'll go to the next slide, which depicts a Foodweb. Oh, I'm not sure how this is showing up on your PDF. Does this show up as?
Oh, it might show the just like your second picture there, but
I'm just wondering if it's filled in with.
It might be, yeah.
Okay, so a good soil Foodweb will have a whole bunch of organisms. I'm hoping you're seeing a whole bunch of organisms in rectangles, and it starts on the left side with the plant, because the plant is feeding. It's feeding the soil. It's providing that energy, organic energy, to soil, in often cases nutrients such as nitrogen. And then we have bacteria and fungi, and some little nematode worms that you see in the top left that are feeding on those roots, okay, and decomposing residues. This is very important. That's the base of the soil biological activity, is bacteria and fungi. These things that organisms that are degrading, feeding on plants. They could be live plants in terms of being like a herbivore, or they could be decomposers. Okay, they're very important. They make up the bulk of the soil organisms. Okay, and then we have organisms that will feed on those. Okay, we have like a tardigrade in the very top there, in the second column, or a water bear. If you're a fan of Star Trek Discovery, the tardigrade is in space. Okay, the mycelial network in space. Then there's the springtail below that, and other nematodes that feed on fungi in the middle there, and bacteria at the bottom, protozoa, and so forth. And you can see it goes up to bigger organisms as we move to the right. Okay, because they're feeding on things below to the left. Okay. That's a healthy food web. We need this because the service of this food web is to provide nutrients. Okay, once they're feeding on organisms on their prey, they will be sloppy eaters, and they will also, what I refer to my students, I tell them they poo and pee just like us, and when they poo and pee all these organisms, they're releasing nutrients into soil for other organisms and for plants. we want that. If you go to the next slide, a poor soil in terms of health is one with a very truncated food web. Okay, we start losing out on those higher trophic organisms. Okay, those say earthworms and foul bugs and so forth, carnivorous nematodes. We lose out on them, and we still have a lot of bacteria and fungi. Actually, we probably even have more bacteria and fungi in an unhealthy soil because there's less things eating them in terms of predators, okay, on them, so their population remains high, okay? And this is a problem because as soon as we start having this unhealthy food web, then we start losing out on the services provided to plants, okay? And so when we do testing of soil, it is possible to examine soil for the different organisms to talk about health. It's challenging though to have a commercial test for that. In my research laboratory we can do this, but it's unfeasible to give you, at least at the moment, in the future, and the future is probably going to happen faster than I think, you will be able to get quite a comprehensive view of soil organisms because of the molecular DNA technologies that are coming up. Okay. All right, I'm just gonna unmute everybody and check in for questions.
The conference has been unmuted.
Any questions or comments?
You mentioned about the idea of the importance of continuous testing of soil. How important do you feel it is to have the exact location down to the GPS points retested over time, that variability within fields? How critical is that in a management perspective at a field scale?
That's super.
Yeah, that's
another place for
that's a great question. So, do we have to resample at the exact location? Well, ideally, we want to go back to a similar area. If it's the exact location down to the inch, that's not good because we're not going to be encompassing field variability. So what I recommend is multiple locations in a field that are tested. And so what I like to do is set up benchmarks. So you take, let's say you're in a field, you take 6, 8 spots in a field in terms of exact locations, and then sample within a radius of, let's say, 2 to 3 meters of that location. Take a composite soil, sample within that radius, and that becomes the sample for that benchmark location. But you have multiple benchmarks in a field, okay? Now I know that increases your costs in terms of having multiple locations, but we really do need that to assess the variability. If we just take one composite sample for a whole field, your chances of seeing changes and improvements is going to be a lot less. You're going to have to wait a lot longer to see those differences, okay? Because the differences are going to be kind of blended out and muted, okay? Does that help? Does that answer the question?
Absolutely does. Thank you.
Awesome. Any other comments? All right, let's go back then to the PDF. Okay, we're gonna go back to muting everybody, okay?
The conference has been muted.
All right, so now let's look at some examples of soil organisms and the impact of management. So this is results of a study by a graduate student of ours quite a few years ago, and looking at the Glen Lee crop rotation and management study, the long-term study led by Dr. Martin Ensign, Plant Science. Okay, sometimes we just refer to as the Glen Lee study, or do you have an official name?
Nope, that works.
The Glen Lee study. So Canada's oldest organic conventional comparison for crop production. At this point in time, it was 13 years into the study. So well, we've gone probably twice that now, which is amazing. So, but at this point in time, looking at organic versus conventional in a grain, oil seed, and also with some forages, perennial forages in a rotation, and looking at grain-only rotation and a forage grain rotation where we had alfalfa present for 2 years within a rotation. Okay. And we're looking at mycorrhizal fungi. So these are fungi in soil that assist plants in acquiring phosphorus. These are fungi that have evolved with plants on land. And we think that they're actually critical for the colonization of plants on land in evolution. So these are some fundamental organisms here. And we can see that they respond due to management. The first thing that you can see is the column that says ARBCOL. That's arbuscule colonization. So these are structures of the fungus that are really assisting in phosphorus transfer to the plant from soil. And you can see in all cases in the organic system, the colonization in terms of these useful structures produced by the fungus to transfer phosphorus to the plant are always higher, consistently higher. It's pretty incredible. And the total colonization is all the structures of the fungus and the roots. Again, higher with the organic. Okay, this is very interesting, and it tells us that in particularly, what is it about organic systems that is spurring on this association with this fungi? Well, it primarily is phosphorus. The phosphorus in the soil was lower in the organic systems, and as a result, the fungi were, the plants were relying, the crops were relying more on the fungi to acquire phosphorus. Okay. Quite interesting. In the conventional system, the phosphorus levels were fantastic. They were what we would refer to as adequate to borderline, not super high, but more than enough phosphorus. Okay, they're around 23, 24 ppm Olsen P in the conventional system. Whereas in the organic system, we're talking about down to 6 to 9 ppm. Okay. So it's a good example here of our management impacts on the organisms. The next slide, which is an example of tillage and rotation and phosphorus, is work done with wheat and looking at flax and canola preceding the wheat, the spring wheat. We did this for 3 years, and we also looked at tillage in terms of conventional tillage and a minimum tillage, so still some disturbance with the seeding though. And let's look at the tillage. Over the 3 years, we can see that the minimum tillage had higher colonization of these fungi in the spring wheat. This is another thing. The physical disturbance, so the mixing of soil, is very impactful on soil organisms, particularly these fungi, because these fungi produce hyphae, which are like, I call them little highways. They're highways. It's a infrastructure network of fungal strands that can move and translocate phosphorus from areas in soil where the phosphorus is located to the root system. Well, if we go in and mix up the soil, we destroy that infrastructure, then the fungus has to rebuild it. Well, that takes time, that takes energy, and so forth like that. It's not going to be very efficient for the fungi. Okay. The preceding crop, in terms of in 2 of the 3 years, we can see that flax increased the colonization of the spring wheat compared to canola. This is very important because This is really important because canola is non-mycorrhizal, okay? And I'm just, hang on a second here, I got somebody that just texted me about, okay, let me go back here, sorry, I lost my train of thought here. Oh, sorry, one moment. Oh, I think that's for something else.
Okay.
Yeah. Okay, sorry, I'm back here now with you. Canola is non-mycorrhizal. So anything in the mustard family, as many of you know, will not associate with this fungus. So the crop like spring wheat following canola will have reduced levels of colonization, whereas flax really loves mycorrhizae fungi. Actually, it's probably, in terms of our crops, probably the plant that most needs mycorrhizae fungi. And they will actually bump up the levels of mycorrhizal fungi in soil, and crops after flax will benefit from the mycorrhizae fungi. Okay. So this is very interesting as well. So you can actually use flax in a rotation to bump up the mycorrhizae fungi. If we can avoid the mustards, then we can keep the levels high for the mycorrhizae. Okay, next slide then, example of canola reduces mycorrhizae and yield. Now this is work that was done in Australia, and what we have is yield on the y-axis, and on the x-axis we have percent arbuscular mycorrhizal fungi. These are mycorrhizal fungi. Okay, and you can see this relationship of that curve, or that line. It goes up in terms of yield with more colonization of arbuscular mycorrhizal fungi. And each of those points, there are data points in fields, and the previous crop to wheat is shown there. And one of the things you see is the lowest yields tend to be crops, where canola was grown the year before. Okay, you see that? You say like the two furthest left points, canola hyola and canola narindra. Those are different hybrids of the canola, and there's even fallow in there, and even those two fields were lower than the fallow field. So there is an impact on our choice of previous crop. In this case, the phosphorus was probably pretty low in these fields, and therefore you really see an impact of the mycorrhizal fungi in low testing soils. So if your soil is testing something like, let's say 10 ppm Olsen P, then your management for mycorrhizal fungi is probably going to make a big difference on your yields. Okay. Let's go to the next slide. This is an example of bacterial communities. Again, a study from the Glenlee experiment, just south of Winnipeg here. And this is for bacteria. Now this is a fancy diagram. The key thing I want you to look at is the reds and the greens. The greens are all treatments that are in organic production, and the reds are in conventional management. In this case, meaning herbicides and fertilizers. There might have been fungicides. Do we apply for wheat? Do we put any for fusarium? Yeah.
I'm not sure.
Yeah, I'm not sure if we're using doing any fusarium control on wheat in the conventional studies. What this method was doing was looking at the bacteria, extracting the DNA from soil of the bacteria, and then so we have in here hundreds of different species of bacteria that make up each single data point here. Okay. And what it does is it kind of groups the bacterial community in a different place on the figure, okay? And you can see the grouping of the organic management systems separate from the reds of the conventional, which is telling us those communities are different in their species composition, very big impact in terms of management is evident here, and this was probably after 22 years of management, okay, in terms of being organic or conventional, okay. Next slide, a summary of nematode analysis for Manitoba soils. This does a similar thing in terms of plots up, in this case now not bacteria but nematode communities in soils, and you can see quite a difference in potatoes, rotations with potato. Other crops seem to be shifted to the right in their positioning on the figure. And then when we go to prairie, those are native prairie, native prairie, and also reestablished prairie at the Glen Lee study is in there as well. And then we have tame hayland or forage perennial grasses with and without pig manure, and you can see the pig manure jolts it up into a different location. And what we're seeing here is that anything to the left is really based on the nematodes communities, is telling us that the soil is unhealthy relative to anything to the right. Okay, so those prairie sites are much healthier in terms of soil food web. Okay, there's higher, there's larger soil organisms, higher trophic levels, there's more diversity and so forth like that. And then you can even see other crops. So these would have been wheat and cereals and mostly cereals, and we'll probably even canola was in the rotation. And you can see they're to the right of the potato rotations, okay, those squares and circles that are colored, okay. And so it's interesting. So what is it about potato? Well, think about it. We're sieving soil, basically, right? We're passing soil through a screen when we harvest potato. And we're growing potatoes on very light soils that do not have much clay and organic matter and so forth like that as well, okay? So those soils look less healthy than soils in other crops, and particularly to prairie grassland. Next slide. More example, nematode communities. This is at the Glen Lee study as well. And this really shows the impact of restoration of agricultural land to prairie. Those are those triangles on the far right. Again, these are about nematode communities. And moving to the right tells us that we're having a shift in the nematode communities, which indicates a shift in the food web to higher trophic, more diversity organisms, okay? It's not dominated by bacteria and fungi, that food web, okay? Whereas we go all the way to the left, we see more of the conventional, the open circles, and the organic, we can see a little bit of a shift to the right in the organic, which is the dark filled circles, okay? So a small shift going to organic, but a monster, shattering, drastic change going to the prairie. And it really tells us about the fact that we have a perennial that we're growing. There's a few different species in that prairie, and there's lack of disturbance. We're not tilling that prairie. So this is interesting. I really like this study because it tells us about what do we expect in terms of shifts, okay, between, let's say, our management, such as organic and conventional. Well, that is a subtle change compared to doing something drastic like changing the system, the production system, going to perennial native grasses. It's quite a drastic change. So both the conventional and the organic here still have tillage, okay, and they're still growing the same crops. And the difference in the microbial communities is slight, but there is a difference though, okay? It'd be wonderful to try to mimic that restored prairie nematode community in our conventional and organic systems. That would be phenomenal. Soil testing and the Cornell Soil Health Test. So several tests I just want to bring out here. We're not going to go over them in quite detail, but these are things that I'm sure you've heard about, particularly the Cornell one. That's pretty, it's kind of the leading test at the moment, okay? A slew of properties that are measured, again, they're biological, chemical, or physical properties of soil, okay? And these are done by commercial laboratories. You can submit samples. Really popular in the United States, okay? Just go to the next slide in terms of a test report. This is an example here where we have the categories: physical, biological, and chemical, okay? And the idea is that there's a test value that is provided, and then there's a rating. So where you see the yellow, red, and green. So there's an interpretation done by the testing lab that says, well, green is good, yellow is intermediate, and red is bad quality, okay? It's a bit subjective, okay? It's a bit subjective. I'm not a big fan because we haven't done calibration of the test for our soils on the prairies. But one thing is that you could be using the values and the ratings over time. So if you repeatedly sample, let's say every year, and seeing if your soils are changing in the right direction, then I think that is useful, okay? But just getting one analysis without us doing a lot more work in terms of the interpretation, I think is suspect to giving us a false sense of saying our soils are good or bad, okay? So chemical, biological, and physical tests, these are pretty straightforward tests, a little bit of a mix of traditional soil testing in terms of chemical analysis for nutrients, and then there are water availability, soil organic matter, and so forth in there. So I'm sure some of you, maybe even some of you, have actually done or gotten analyses of the Cornell soil health test. The next one is the visual state assessment test. This is now more for perennial-based systems, particularly grazing systems from New Zealand where they have lots of dairy. And so this was a soil health test done really for looking at which emphasizes physical structure of soil, okay? And something you can do yourself. You don't have to pay anybody to do this, okay? And let's go to the next slide, which shows a scorecard and score sheet. And it really talks about the structure, the porosity, the color of the soil, earthworm counts, and so forth like that, okay? And it's really about the physical aspects of the soil. There's no chemical analysis being done here. And it's really what it's saying is that, hey, if our physical properties improve, soil health is improving. And in this case, it makes sense because we have forages that are perennials, and that root system will be building up that structure and then improving the physical properties, okay? And so that's what the reliance on here is. The next slide shows in terms of the structure, going from good on the left, which would be a crumbly soil, all the way to the right, which would be a non-structured soil, just big clods that would indicate extremely poor health. Okay, and we know the benefit of roots in terms of getting us that crumbly structure. It's one of the reasons why we would like to try to reduce tillage in our systems. The next one, in terms of soil porosity, in terms of slide, and here you can really see the roots. And the crumbly nature on the left, you can see lots of roots in there. The soil is crumbly. We go to the right, it's just a lump of clay. Kind of looks like the Red River Valley sometimes, right? Once it dries up, it'll be darker, be black. But you can see the lack of roots. There's no crumbs in there. Imagine if you were a root. You're not going to be able to move through that poor condition soil on the right there. Okay. Another test is looking at PLFA, or phospholipid fatty acid. This is a specific test which looks at groups of organisms in soil. Okay, so it's done to generalize about the soil organisms in soil. It particularly generalizes about major groups of bacteria. It doesn't do species of bacteria, but just does some major groups of bacteria and total fungi in soil. And so this analysis is being used to look at bacteria and fungi, particularly the relative amounts of bacteria and fungi in soil. So some test reports will give you a bacterial to fungal ratio based on PLFA, okay? And the next one is the Haney soil test. We had a question about that earlier on. And this one is very interesting in that it focuses on the chemistry of soil and particularly the nutrients in soil, plant available nutrients. I'm personally not a big fan of this test because it really says, well, it's mostly nutrients in soil that are really critical. And I'm not sure about that. I think the biological function of soil can really help out with the nutrient acquisition by plants. And I think the Haney test doesn't give that credit to organisms and soil physical properties, okay? And you can see this is an example from AgVise in terms of a Haney soil test. And look at there's a slew of extractions here. A lot of these extractions actually we do in our research, okay? And it gives you for all the macro and micronutrients, and then talks about, breaks down, say, nitrogen and phosphorus to available forms of that nitrogen and the ability for mineralization of nitrogen and phosphorus in the soil. So some utility about that. But I find this a little bit challenging. This test was developed for Texas. It was developed in Texas. And I don't know how you interpret this if for our soils on the prairies. It needs work to be done to do that interpretation in terms of what the levels are and what it means for our crops. We can't rely on the work that's been done in Texas to do that, okay? So anyway. Another one here, the Woods End Laboratories. Sometimes you hear this is called Solvita Laboratory. And they look at a lot about fertility again, and they do some organic matter, and they look at CO2 or respiration bursts from drying soil and wetting it with what's referred to as the Solvita test, where you put soil in a jar, you put this little paddle, and it changes color based on how much CO2, how much respiration occurs. More respiration is assumed to mean more biological activity or health of the soil. Okay, this is okay, but from a nutrient perspective, I think it's pretty decent. It gives you basically what sending a soil sample off to a conventional lab would give you. But this is available as well.
About 5 minutes.
We've got 5 minutes left, I'm told by Catherine. And then there's a soil Foodweb analysis. This was pretty hot about 15 years ago. I hear a lot less about it now. This one really looks at organisms in soil. Okay, bacteria, fungi, protozoa, nematodes, and so forth. And a lot of the analyses that we do in the lab, my lab here, for in our research. Okay, it's interesting, but I am not sure how these are useful though for farmers. In terms of, again, the interpretation of the results needs to be done for our soils, and that work hasn't been done. Go to the slide that says what can be done to improve soil health, okay? All right, and so there's a couple of things here. Rotation. So we're talking about rotation wisely, which would be not relying on canola, and make sure we have a diversity of crops. That could include polyculture. It can include cover crops. Okay, those are, I would consider that increases the plant diversity, and that's going to be a good thing for soil health. Increasing soil organic matter in soil is probably the most important thing that you can do to soils, okay, to improve its health. And how do we do that? Increase the biomass of grow high biomass crops, try to include perennials, reduce tillage, use cover crops, use animal manures or compost in terms of addition, okay? So just get more organic matter, feed more of your soil, okay? Feed it better. And then a thing that we often overlook in terms of feeding the soil is roots. Roots are becoming more and more, in terms of our understanding, it links better to increase in organic matter than above ground crop residues, okay? So plants that have rooting systems that are large and that turn over and die often or multiple times in a growing season can increase organic matter way more than growing one crop and putting in a whole bunch of residue and plowing it into the soil, okay? Grow your soil, so having a mindset saying, I'm not just growing plants, I'm growing soil. So think about, over time, I need to be increasing my organic matter. I want to see that in soil tests, for example. I want to see a change in color. I want to see a change in aggregation when I take a shovel of soil, okay, out of my fields. So you want to see an improvement, okay? Go to the next slide. What can you do to improve soil health? The second slide. Test for nutrients in your soil. Not just before the crop, but after the crop, okay? Because see if you have a lot of nutrients that are left over, okay? That would mean that we have a problem in terms of we're providing too many nutrients. We could cause environmental issues. So particularly with organic systems, I would say look at your nitrogen. Nitrogen through the roof in terms of nitrate at the end of the season. You might be doing a great job managing nitrate and maybe too well in terms of getting nitrogen in high levels, but have poor phosphorus, for example, okay? So I think sometimes we may really strive to get nitrogen into our crops and actually have way too much of it present after the crop, okay? So get a soil test done for nitrates. Test for organic matter, okay? If you test for organic matter, do it from the same depth, over the same depth, and do those benchmarks that we discussed, okay? Look for worms in your soil. Okay. And then repeat tests over time, okay? Track it, okay? Track and see how you're doing. Are you building equity in soils, such as organic matter, okay? So we just had a comment about, for me to mention about, is it worthwhile to inoculate your soils with, let's say, for example, commercial bacterial inoculants that we can buy? Does it improve soil health? At the moment, there's not really good evidence of an improvement by adding inoculants in terms of general bacteria. There is demonstrated, of course, for nitrogen-fixing bacteria with our legume crops. Absolutely, for sure, that's documented. But in terms of other bacteria that are out there, there are various. There are pseudomonads, bacillus. There's a whole bunch of different bacteria out there, and there's some fungi as well. At the moment, they're not, at least what I can see, they're not really useful. And one of the things is that the organisms are already resident in your soil. They're already there. And it's just a matter of our management to bring them out to be effectual in soil, okay? So by adding them to soil doesn't really get at the issue here. The issue is the management rather than the presence of the organism. The management will change, will encourage the organism to have an effect, okay? It's kind of like if you build it, they will come, kind of thing. If you build your soil, the organisms will come. You don't have to add them, okay? Now, having said that, I'm going to throw a wrench into our thinking here, okay? And that is, we have now some of the largest companies in agricultural input industry now owning divisions and selling bacterial and fungal inoculants or biologicals, okay? And so these are some large companies. And I do think once they throw in research dollars, that in 10, 15 years from now, I may be saying to you, hey, these are wonderful products, okay? So I don't want you to think that they're going to be useless down the road, okay? They may very well be, and I think if they throw in great research effort into it with money, that I'm sure they will provide new inoculants that will be useful down the road, okay?
Great. Is that it? Are you good?
I am good.
Okay. So just for right now, does anyone have a last question they'd like to ask Mario?
I have a question.
Go ahead.
There has been no discussion at all about the role of calcium in flocculation of the soil to increase the porosity and the airspace. Any comments on that?
Okay, so it's a good question about calcium. Calcium is very important in soil. As you're saying, it really improves aggregation. Calcium is useful if you have clay. You need to have clay, and then with calcium you can have improved aggregation. Okay, so if your soil is with a low pH, and I'm talking here below 6, then likely you're low on calcium as well, because that's going to be released from the carbonates and various other things and out of the soil. But so if you have a low pH soil with clays, then probably adding calcium will improve the aggregation. Okay, and the other thing about calcium, what it does is it allows for organic matter to build up in the soil. Okay, because it provides a bridge between the organic matter and the clay. So now, in other words, it allows the organic matter to stick to clay, with the calcium being a little bridge between them. So calcium is important, and I think there are situations in our soils that we can improve the organic matter and the structure with addition of calcium.
Just to follow up on that, how about on the higher pH soils where magnesium is driving up the pH? Can we use calcium to try and get a better balance in the cation exchange capacity between calcium and magnesium?
Okay, well, magnesium itself will not drive pH, and calcium itself will not drive pH. It's what they're associated with, and oftentimes they're associated with carbonates in our soils. So dolomite for magnesium and calcium with limestone or calcite. So magnesium itself won't be affecting the pH, but it's what it's associated with. So magnesium will do the same thing with as calcium will do in terms of building organic matter and improving soil structure. They're both what we refer to as divalent cations, so they will function similarly in terms of improving soil health. Okay. Now I do understand what you're mentioning about a ratio. Is there is a train of thought or a line of thinking about the ratio is important in terms of nutrition for plants. That aspect, I haven't done that, examined that in my research per se. I have looked at some of the research in the literature regarding to that, and I must admit I don't follow the argument of those ratios. Okay, I would put my attention in other places in terms of improving soil health.
To thank everyone for joining the call, and thanks again to Mario for taking the time out of your day